In this series of videos, I'm going to talk about Chapter 8 in your textbook, the control of movement. I'm going to break it up into 6 different videos. I'll spend about half hour or so talking about anatomy and innervation of muscles, spend a few minutes reviewing the neuromuscular junction, a little bit of time talking about the physiology of contraction, talk about reflexes, how the brain controls movement, and then mirror neurons. One thing I want to point out as I go through this, I'm going to be simplifying quite a bit. The textbooks that cover movement for neurology programs are 600, 700, 800 pages long. It's incredibly complex. So we're just going to scratch the surface of it. But obviously psychology is the scientific study of behavior. Movement is a key part of almost every behavior. So in order to understand psychology, we do need to understand a little bit about movement. In this video, I'm going to cover the anatomy and innervation of muscle. After you watch this video, I'd like you to be able to describe the basic structure of muscle, explain the innervation of a muscle, and talk about some of the changes in activity you would expect to see in the sensory neurons that innervate something called the Golgi tendon organ and the muscle spindle. We'll cover those soon enough. Before I get into it, I need to cover some general terms. These aren't things necessarily for you to memorize for the exam, but without these you'll have a tough time understanding what's going on. So memorize what you need to understand. Right. In this case, the book is going to use the biceps brachii muscles as an example. Okay. This is going to be a skeletal muscle. That means it's under voluntary control. It moves when you want it to. It's going to be located in your upper arm. Now, if you look at the structure, we're going to have this nice big belly. That's a bulk of the muscle. So when I flex muscle, you get that nice big bulge, right? That's the biceps brachii. Now all muscles, nearly all muscles, are going to be attached to bone through tendons. Okay, so that occurs at each end of the muscle. Now muscle is stupid. It can do 2 things, and that's it. It can contract and it can relax. When muscle contracts, it gets shorter. Okay, so in this particular case, when the biceps brachii contract, they get shorter, and that's going to pull the ends together. That's going to result in flexion of your arm. When that muscle relaxes and a muscle called the tricep on the other side here contracts, our arm is going to straighten out. Okay, in this case, that's going to be called extension. Okay, so flexion is going to be bending the arm. Extension will be straightening the arm. Sorry, flexion will be bending the arm. Extension will be straightening the arm. Okay, so flexion, extension by moving the ends together. Now, when this shortens, you know that it's going to be your lower arm that moves up. This is your shoulder. This is on one of the arm bones, the ulna. So when this contracts, this end moves up. Okay, so there's 2 ends to a muscle. One end is called the insertion, the other is called the origin. When the muscle contracts, it's the insertion that moves. Okay, so up here in this case, we have the origin. Down here, we have the insertion. Now, the tendons aren't that strong. They're pretty strong, but they're not incredibly strong. Most of us are actually strong enough to rip the tendons off the bones by contraction of the muscle. We need to prevent that from happening. In fact, there's a reflex that'll help us do that. I'll talk about that in a minute. For right now, I'm just going to talk about the anatomy that's relevant to that. And that's going to be the structure called the Golgi tendon organ. You can see that this GTO is going to be part of the tendon. When this tendon gets stretched, we get increased firing. Okay, so stretch equals increased signaling to the spinal cord. Okay. Now, when we come over here and look at the muscle itself, you'll notice that there's going to be two different types of muscle fibers. One is called the extrafusal muscle fibers. The other is called the intrafusal muscle fibers. These extrafusal muscle fibers generate force. Okay, so when these are stimulated and contract, I move things. These intrafusal muscle fibers are going to be in this thing called the spindle. These intrafusal muscle fibers are primarily sensory. Okay, these are also stretch receptors. So when this muscle fiber is stretched, I get increased signaling. Okay, and when it's not stretched, I get decreased signaling. So when I contract my biceps brachii, what happens to the rate of the signaling in this intrafusal muscle fiber? Well, when I contract, it's getting shorter, right? It's not stretching. So as I contract my biceps, I actually get less signaling. Now, when I relax my arm, the muscle is starting to stretch. These intrafusal muscle fibers stretch, and that's going to cause them to be stimulated. Okay, so I'll get that increased signaling. Now, there's sensory neurons. Okay, you can see sensory endings here. These sensory endings are going to be what detects and then carries that information about stretch back to the spinal cord. Okay? So the intrafusal muscle fibers, I want you to think stretch. The extrafusal muscle fibers, I want you to think force. Okay. Now, I told you that the intrafusal muscle fibers are going to send sensory information back to the spinal cord. So there's a sensory neuron involved there. There's also going to be efferent neurons coming from the spinal cord. And there's going to be two different types. There's going to be alpha motor neurons and there's gamma motor neurons. Alpha motor neurons innervate extrafusal fibers. When these alpha motor neurons are excited, here I'm going to abbreviate, action potential equals contraction. And you know that these extrafusal, right, this equals force. Okay, so when an alpha motor neuron is stimulated, the action potential comes to the terminals. I get the release of neurotransmitter. That's going to cause a cascade of events that results in contraction of the extrafusal fibers. Okay, so alpha motor neuron stimulation equals muscular contraction. With the gamma motor neurons, this is going to send action potentials down to the intrafusal muscle fibers. It will cause them to contract. But as I said before, even though these contract, contraction equals no force. What it does is alter sensitivity to stretch. And I'll talk about how that works for a minute, in a minute. So let's imagine a disease where all the gamma motor neurons died. Could that person still contract their muscle? Yes, they could, because the motor neuron, right, the alpha motor neuron, would still be able to cause contraction of the extrafusal muscle fibers. What happens if I block alpha motor neurons but not gamma motor neurons? Now in that case, I'd have paralysis, right? I wouldn't be able to contract. Now, just as an aside, if we did get rid of all the gamma motor neurons, there would still be problems. We'd be able to contract, but movement would not be normal. You need those. Okay, so that's the innervation. We've got sensory neurons going to the Golgi tendon, sensory neuron going to the muscle spindle. That muscle spindle is the intrafusal muscle fibers. Same thing. Okay, so those would be two sensory afferent neurons. We also have two efferent neurons. We've got the alpha motor neurons and the gamma motor neurons. Now, when we look at a muscle and we look at these extrafusal fibers, those bundles are actually made up of individual muscle fibers. Those are just like long threads. Each of those muscle fibers is going to be made up of a myofibril. Okay, so here's a muscle fiber made up of a whole bunch of individual myofibrils. Here's a myofibril. Now with the myofibrils, you can see this banding. Okay, so skeletal muscle is also called striated. Striations, stripes. Okay, and that's going to have to do with the arrangement of filaments. I'll show you that in a picture in a minute. Right, but the myofibril is going to be made up of actin, myosin, and a couple other things. But really for this class, actin and myosin is what I want you to remember. Here is another diagram showing it. Usually I'll show you a couple diagrams just so sometimes one clicks better than the other. Alright, so here I've got a skeletal muscle fiber. In this case, that's going to be an extrafusal. Here we've got one myofibril. The myofibril is going to be made up of repeating units called a sarcomere. And so the sarcomere is the basic unit of contraction. When you look at the sarcomere, you're going to have these thin, here they're blue, that represents actin. That's already labeled for you. And then here, these thick, it's going to be myosin. Okay, so myosin is thick, actin is thin. These two things are going to interact, and that's going to cause contraction. The reason you have striations is if you look here, for example, actin and myosin don't overlap. There's nothing really there. So that's going to appear light. Here, though, if you look, I've got actin and myosin overlapping. So that appears dark. Here, I've got myosin only. Right? So it's going to appear kind of light, except for the bump in the middle here. It's a larger part of the structure that you can actually see. And then it repeats. So here we've got that overlap again, so we get dark, and then here we get light. You don't need to memorize that. I don't want you to know A band, I band, H band, all that stuff for the exam. But I do want you to know that these filaments are going to overlap, and it's that overlapping that makes for the striations you see in muscle. Now, when we look at myosin here and actin here, there's a couple things you're going to need to remember when I get to contraction in order to understand how it works. On actin, we're going to have a protein called troponin and a protein called tropomyosin. Okay, so you need to know that those are on actin. In terms of myosin, I just want you to know we have these big cross bridges. To make a long story short, these cross bridges are going to interact with the actin and essentially pull the actin toward the middle. And when it does that, obviously that's going to shorten the sarcomere and we'll get muscular contraction. Okay, but for now, we're just going to go over, I just want you to know the structure. Here's another diagram showing you the same thing. Okay, so if you want, you can pause and look at this for a minute. It's just another way to give you an idea of the basic structure. Now, later on, I'm going to talk about the reflexes a little more, but I'm going to introduce it here. Something called the stretch reflex. Okay. Right now, we're talking about the intrafusal fibers. Okay, so this is really the muscle spindle. Now, when a muscle relaxes, it's going to stretch. Okay? That's going to increase afferent signals to the spinal cord. In the spinal cord, that's going to stimulate alpha motor neurons. Now, when alpha motor neurons are stimulated, what happens? We get muscular contraction. That's going to shorten the muscle. When we shorten, what happens? We get a decrease in the firing rate of those afferent neurons that go to the muscle spindle. And this is a typical negative feedback system. Okay. Now, we can look at it another way. We can look at muscle length and action potentials in that spindle sensory. Before, when the muscle is just sitting, before we start anything here, okay, you'll notice that that spindle sensory neuron has some spontaneous activity. I've told you many times, neurons are always active. There's always some spontaneous activity. And really what the nervous system's sensitive to is changes, increases or decreases. All right. So now here, when my muscle is stretched, what's going to happen? Well, now it's going to get stretched. We're going to get increase in firing rate. Okay. The greater the rate, remember the rate law, the greater the rate, the greater the stretch. Now, this sensory neuron is going to increase its firing rate until the muscle returns to its initial length. Okay? So, as long as the muscle is long, we're going to get increased firing. When that happens, right, we're going to get stimulation of the alpha motor neurons and muscular contraction. Now, I'll talk a lot more about this when I get to reflexes. We'll talk about the anatomy a little bit more detail, and we'll talk about the function of it. One place where you do see this is in the patellar reflex, also known as the knee-jerk reflex. In the knee-jerk reflex, right, the doctor hits your patellar tendon. Okay, now you're likely to think about Golgi tendon organs. Don't do that. That's not what it's doing right now. When you hit that patellar tendon, it actually stretches these fibers. So when those fibers stretch, what happens? Yep, alpha motor neurons increase in activity, and I get muscular contraction. Okay, so I hit that patellar tendon. That actually stretches these intrafusal, the muscle spindle, in the quads. Those quads then contract and cause your leg to extend. All right. Now, with the Golgi tendon organ, I also talked a little bit about this. I told you that Golgi tendon organs are going to detect tension that are applied to a tendon. Okay, so essentially, imagine I'm trying to pick up something that's too heavy for me. As I do that, my muscle's going to contract. Okay, and as my muscle contracts and contracts, it's going to exert force on those tendons. And if my arm doesn't move to relieve that tendon, right, eventually that force is going to get so strong I can rip my tendon off my bone. Well, what happens is sensory neurons linked to that Golgi tendon organ, right, are going to bring afferent signals into the spinal cord. Here, they're going to stimulate an inhibitory interneuron. Okay, so when this neuron is activated, it's actually going to cause inhibition of the next neuron, in this case an alpha motor neuron. So excitation of this leads to increased inhibition, which means now this alpha motor neuron is firing fewer action potentials. When that happens, now I get relaxation. Okay? Now this has a couple benefits. We'll talk a little bit about those later, and I'll talk more about this reflex later. Once again, a quick overview. We've got the Golgi tendon organ, right, sending sensory information to this interneuron in the spinal cord. That's going to inhibit the motor neuron. Now let me ask you real quickly. Remember, the muscle spindle, we'll learn, is monosynaptic. So that's going to bring right in, and it's going to synapse right onto the alpha motor neuron. So in this case, which of these two, this one or this one, do you think is faster? Now make no mistake, both are really fast, right? But fewer neurons equals quicker response. Okay, so it turns out this, mediated by the muscle spindles, is faster than this Golgi tendon organ reflex. All right. So again, I talked about the muscle spindles and the GTO being innervated and what stimulates them. So let's imagine I've got my arm flexed, and I'm going to slowly lower it and extend it. Now let's think about this. If I do it slowly, start the movement here, what happens to that GTO? Is there increased tension on the tendon? If it's slow, there won't be any. Okay, so we'll get no change in activity here. Now here we'll have two different muscle spindle. Okay, this represents a sensory neuron innervating two different intrafusal, right? These are the intrafusal muscle fibers. Two different spindles. Slowly lowering the arm, what happens? Well, I'm relaxing it, right? I'm extending it. So we are going to get some stretch. The key being slow and passive. Okay. So in this particular example, one of the muscle spindles will react and increase firing. The other has no response. Okay, so in this case, we do detect the stretch, okay, but it's very mild response. Now let's compare that where the arm is abruptly dropped. Okay, so you're holding it still, and then suddenly you move down. As you do that, is there increasing tension on the tendon? If the muscle's relaxing and my arm goes down, obviously there's none, right? Okay, so the tension on the tendon stays low, and that neuron that innervates the GTO doesn't respond. Here, if we're doing it quickly though, now both of these spindles are going to respond. Okay, so the stronger the stimulus, we get a faster rate of firing within a neuron, but in addition, more neurons will respond. Now, here you're holding your arm straight. Now, suddenly, a weight's dropped into the arm. So now a few things are happening. When the weight is dropped into your arm, right, initially, your biceps is going to contract to try to counteract that. Now, when my muscle is contracting, what's that going to do now? in that case, right, I'm actually going to put tension on the tendon and I'll start to get a response. Now, in order for me to move my arm back to its original position here, I have to contract my muscle, right? So in this case, when the muscle is contracting, I'm putting tension on the tendons, okay? And that's why the GTO is going to continue to respond as long as the weight's in the hand. Okay, so this sensory neuron that innervates the GTO will be going along at its resting rate. I add the weight, suddenly there's increased tension, and I get increased firing. And that stays that way even after my arm returns to its initial position, because to hold the weight, I have to contract my muscle. Now, let's think about the muscle spindles. These detect stretch. So in this case, here we are at rest. We drop the weight. What happens to my muscle? In this case here, the muscle has stretched. Okay, so when we add the weight, these neurons are both going to fire. Now, when we return to our initial position, the length of the muscle is the same as it was at the beginning. So, here we are, our length at the beginning. While our arm moves down a little bit, the muscle spindle is stretched, we get increased firing, and the time we go back to our regular position, right, we're going to have the same firing rate as what we started with. So, after this video, I hope you could describe the basic structure of a muscle, just what I went over. You don't need to go into much more detail. With the innervation, we have afferent, sensory afferent neurons. I've got one going to the Golgi tendon, or going from the Golgi tendon organ to the spinal cord, one going from the muscle spindle to the spinal cord. We also have alpha and gamma motor neurons. The alpha motor neurons innervate the extrafusal fibers, right? And that's going to result in generation of force when they're stimulated. We have the gamma, which go to the intrafusal fibers, which are part of that muscle spindle. Okay? Those are going to modulate sensitivity of the muscle spindle, but they do not generate any force. In terms of the changes in the activity, that's what I went over in those last couple slides. If you have any questions on this, please do feel free to email me, and feel free to give me any feedback on the videos. Thanks for listening. I'll see you in a few minutes.